Welcome to The Explainer. I am absolutely thrilled to be your guide today because we're about to dive into what is literally the ultimate biological origin story. Think about it for a second. Every single organism, even the absolute largest ones on Earth, starts out as just one solitary cell. It's wild. So today, we're treating this explainer like an intro college biology masterclass. We're going to build the ultimate study guide together, breaking down exactly how that single cell manages to construct complex, multi-million-celled organisms through the incredible step-by-step mechanics of cellular division. Let's get into it. Quick look at our syllabus for today. First, the cell cycle journey. Second, mitosis, the equational division. Third, cytokinesis, splitting the cell. Fourth, meiosis, the reduction division. Fifth, a mitosis versus meiosis comparison. And finally, a biology professor practice quiz. All right, let's kick things off with section 1, the cell cycle journey. So I really want you to keep this hook question in the back of your mind throughout this entire explainer. How does one single cell become a complex, multi-million-celled organism? Well, the short answer is that both growth and reproduction are fundamental characteristics of cells. Basically, every time a parent cell divides, it gives rise to two daughter cells. Those cells grow, they divide, and cycle after cycle, that one single cell proliferates into a massive population. But it's not just some random splitting, It's a highly coordinated sequence that we call the cell cycle. Now, this cycle really has two basic phases. There's interphase, where the cell prepares itself, and then the M phase, where the actual division happens. Interphase is broken down further into the G1 phase for cell growth, the S phase for DNA synthesis, and G2 for protein synthesis. In human cells, the whole cycle takes about 24 hours. But here is a totally mind-blowing fact. The actual M phase division, the physical splitting, only lasts for about an hour. The interphase preparation steps take up more than 95% of a cell's life. It's kind of like a grand stage production, right? The performance goes by in a flash, but the backstage preparation takes forever. During that critical S phase, the amount of DNA literally doubles, though the number of chromosomes stays exactly the same. And because this cycle is so powerful, medical treatments like chemotherapy specifically target these rapidly dividing phases, acting as a checkpoint to stop cells from proliferating out of control. But what's really fascinating here are the exceptions. Not every cell is just stuck on this constant treadmill of division. Some adult animal cells, like your heart cells, don't appear to exhibit division at all. Others only divide occasionally to replace cells lost to injury or cell death. These cells actually exit the G1 phase and enter what's called the quiescent stage, or G0. They remain metabolically active, doing their daily jobs for your body, but they stop proliferating. They just kind of hang out in standby mode, only jumping back into the cycle if the organism specifically calls upon them for tissue repair. This regulation is absolutely vital for human health, because errors in this standby phase, where cells divide uncontrollably instead of resting, are exactly what lead to diseases like cancer. Moving right along to section 2, mitosis, the equational division. Okay, so mitosis is divided into 4 stages of nuclear division, kicking off with prophase. If you're looking through a microscope, here are the visual cues you'll need to identify it. Before this, back in interphase, the DNA is just this loose, intertwined mess. But in prophase, that chromosomal material untangles and condenses into distinct, compact mitotic chromosomes made of 2 chromatids. Meanwhile, the cell's machinery starts kicking into gear. Centrosomes head to opposite poles of the cell, radiating microtubules. And to make room for all this movement, the cell completely breaks down its internal walls. The Golgi complexes, the ER, the nucleolus, and the nuclear envelope, poof, they simply disappear. Now, to truly understand how these chromosomes move, you kind of have to treat the cell like a microscopic machine. As that nuclear envelope completely disintegrates, we enter metaphase. The centrosomes at the poles are shooting out spindle fibers. But what exactly are they grabbing onto? Well, that is where kinetochores come in. These are small, disc-shaped structures on the surface of the chromosome centromere. Think of them as precise mechanical attachment sites. The spindle fibers hook onto these kinetochores from opposite sides, creating a highly logical mechanical push and pull system. You can clearly see here how during metaphase, the spindle fibers have pushed and pulled the chromosomes until they're perfectly aligned right down the center, which we call the metaphase plate. Then, at the onset of anaphase, boom, the centromeres split simultaneously. The spindle fibers shorten, and those sister chromatids, which are now officially considered individual daughter chromosomes, are aggressively dragged away to the opposite poles. If you look closely, you can even see their arms trailing behind them in a V shape as they get pulled.
Eventually, in telophase, they reach the poles, decondense, and a brand new nuclear envelope forms around each cluster.
Okay, section 3, cytokinesis, splitting the cell. So this physical splitting process actually looks very different depending on whether you're looking at a plant or an animal cell. For animal cells, picture a flexible balloon. A cleavage furrow appears in the plasma membrane and gradually deepens, pinching the cell inward until it just simply splits into two. Easy enough, right? But plant cells are encased in a rigid, inextensible cell wall. They literally can't pinch. So instead, they undergo a kind of interior construction project. A precursor called a cell plate starts right in the center of the cell and grows outward. It's like building a brick wall from the inside out until it meets the existing lateral walls. But what happens when a cell divides its DNA perfectly, creates two nuclei, but somehow forgets to slice its cytoplasm in half? Well, you get this bizarre physiological consequence called a syncytium. It's a multinucleate condition, meaning one cell with multiple nuclei. A fantastic, everyday example of this is the liquid endosperm in a coconut. Yep, all that refreshing coconut water is essentially packed with free-floating nuclei because nuclear division occurred without the physical cell splitting of cytokinesis. Pretty cool, right? All right, let's pivot to section 4, meiosis, the reduction division. Meiosis is highly complex, and that's specifically because of its very first phase, prophase 1. This phase is way longer than mitosis and is broken down into some pretty dense subphases: leptotene, zygotene, pachytene, diplotene, and diakinesis. A great mnemonic device to easily memorize these for your exams is Little Zebras Ponder Difficult Dreams. Let's break down the mechanics. First, homologous chromosomes find each other and pair up in a step called synopsis. Then the real magic happens, recombination. Non-sister chromatids literally cross over and exchange genetic material. It's a precise enzyme-mediated exchange. Later, as they begin to separate, they form these distinct X-shaped separation structures called chiasmata, before finally prepping for separation in a step called terminalization. Now, the absolutely crucial difference here lies in how they separate during anaphase I. Unlike mitosis, where centromeres split and sister chromatids separate, in meiosis I, the sister chromatids remain firmly attached at their centromeres. Instead, the entire homologous chromosomes are pulled apart to opposite poles. This is the exact moment the reduction happens. Each pole is now receiving only half the chromosome number of the parent cell. After this, the cell goes through a short resting phase and then enters meiosis II, which honestly looks almost exactly like normal mitosis, finally splitting those sister chromatids apart. I really want to emphasize the monumental significance of what we just covered. Meiosis is kind of a paradox. It reduces the chromosome number by half, but it does this specifically so that when two haploid gametes fuse during fertilization, the specific diploid chromosome number for that species is perfectly conserved across generations. Plus, because of that crossing over process we discussed in prophase I, meiosis actually increases genetic variability from one generation to the next, and those variations, they are the absolute crucial engine for the long-term process of evolution in sexually reproducing populations. Bringing it all together, let's look at section 5, mitosis versus meiosis comparison. If you're taking notes, this is absolutely the slide to screenshot. Let's walk through this head-to-head comparison. Mitosis happens in your diploid somatic cells, the normal cells making up your body, and its main job is supporting organismal growth and cellular repair. It uses one division to conserve the chromosome number, producing two genetically identical cloned cells. Meiosis, on the other hand, occurs in gamete-producing cells. It requires two sequential divisions to intentionally reduce the chromosome count by half, ending up with 4 genetically variable haploid cells ready for reproduction. Time to test your knowledge with Section 6, Biology Professor Practice Quiz. Question 1. Think back to that comparison table we just looked at. Why is mitosis specifically called equational division? I'll give you a second to lock in your answer. And the answer is because the number of chromosomes in the parent and the progeny cells remains exactly the same. If a parent cell has a diploid 2n number, the two daughter cells are going to have that exact same 2n number. It's an equation that always balances perfectly, ensuring identical genetic complements for tissue growth and repair. All right, ready for question 2? Name the specific stage where centromeres split and chromatids separate. Try to recall the mechanical movements of our microscopic machine. When do things actually break apart? The answer is anaphase in mitosis and anaphase II in meiosis. Remember the visual of those shortening microtubules aggressively dragging the chromatids to opposite poles, with their arms trailing behind them? That forceful pulling apart is the absolute hallmark of the anaphase stages. You all did fantastic. To wrap up, I want to leave you with a final provocative thought pulled straight from our source material. Can there be DNA replication without cell division? Think about it. The S phase occurring, but M phase never firing. We actually touched on the strange consequences of this earlier when we talked about the coconut syncytium. Cellular biology is just full of these incredible exceptions and rule breakers. So keep exploring, keep asking questions, and I really hope this study guide helped clarify the magnificent journey of the dividing cell. Thanks so much for joining me on this explainer.